speaking, and then we'll have nice long question breaks after each part where you can get a chance to ask what questions you have. We will also have an intermission where you can get some more snacks, chat with the speakers, whatever you would like. And yeah, is there anything else you need to add? How things run? Cool.
I think we're ready to get started.
You guys ready? All right, we're going to hook it up. Free microphones. All right. Well, then I'll go ahead and advertise another event series that Science and News runs, which is this other flyer here, the giant beer glass. We have an event called Science by the Pint, which is same idea, communicating science with people, but studying is a little bit different. We bring a professor and their lab to a bar, and they give a short talk, no slides, and then you get a chance to sit at the table, drink beer, and chat with the scientists in the lab and learn more about their research. So if you're interested in that, or if you are 21 years old or older, although actually, younger people can come to the bars, it just might not be quite as fun. We can grab that flyer too. And then we also have a handout with more information about today's lecture and our speakers. So if you didn't grab one when you came in, feel free to grab one during the intermission. All right, all hooked up?
Yes.
Here we go.
Can you hear me?
Yes.
Am I coming through on the other channels or the other things?
As far as we can tell.
Excellent. Well, my name is Lola, and this is, I apologize for introducing myself. This is Wesley and Rebecca. We are members of the Work Lab, and we work on the parasite Plasmodium that causes the disease malaria. And today we're going to give you a lecture that we're titled History's Greatest Arms Race, How Infectious Diseases Have Changed Human Evolution. And just to start, we're going to explain this graphic that you can see that, you know, at the beginning our the attacks and the defenses were rather simple, and the point we really want to get across today is that both organisms have had to change over time in order to continue to survive and continue to grow. So this is sort of a map of where we're going to go today. I will begin by describing the enemy threat and really focusing on the impact that infectious diseases have had on human population and behavior. Then Wes will talk about the arms race, talking about the principles of evolution and natural selection and infectious diseases that really show the back and forth that has happened, and give a really great example. And then Rebecca is going to dive in, really dig into what we call these landmark battles, like these infectious diseases that are still a really big threat for us today, and we'll conclude and look towards the future and the higher ed. So, as I said, I will begin with the impact of infectious disease on the human population. And the place I'm going to start is at with the Black Death, which is also called the plague. I wanted to start with this map because it's one of the most striking things I've seen that really explain the impact of infectious disease on human population. For most of human development, like human civilization, since we sort of like started agricultural societies, we had like, you know, just really trudging along, not having anything major happen, like affect us, even though like, you know, Tom's house was like life is nasty, brutish, and short. But we're sort of holding our own against this pathogen. And you have to imagine the kind of thing that would happen, that there is like a noticeable deep dip in this population curve that's like, oh wow, what's going on here? And what's going on here is a plague. It's a Black Death. It killed so many human beings at the time that it actually like it affected it dipped past the sort of expected level of population. So you have to keep in mind this axis is billions. So that deficit is actually pretty significant. So that's why we started the Black Plague in the 14th century. starts in China. It killed millions of people, and then migrates to Europe, where it actually does a lot of its damage, and 25 million people were killed in continental Europe. Again, keeping in mind global population in the 14th century, that's a pretty significant proportion of people. And this is a gift just showing the spread of the plague across the range of basically 5 to 7 years that is spread all over through from Eastern Europe to Western Europe and just killing a lot of people. As you can imagine, with such death, the sort of like human reaction to it, I really like this etching that shows, you know, the reactions of the common people to it, like what's going on, we're dying, oh, save yourselves, keep out of here, you know, for lots of prayers. This is where the idea of the plague doctor and also the Grim Reaper that is in a lot of history and stories come from this time in our history. So that's just one example of how infectious diseases have really impacted our population and still have like things that are in our cultures and stories till today. So before we go on though, I want to explain a couple of things, a couple of terms that we're going to be just using, like it's ABC, so it's good to explain it at the beginning. And the kind of situation we saw with the Black Death is called an epidemic. And it describes a situation where an infection increases super rapidly and then becomes widespread. And I think that graph I showed you of the plague spreading across Europe over the course of 70 years. And when you think about the number of people that got killed in that outbreak, in that epidemic, that's really typical of what an epidemic is. And we'll have examples of more epidemics as we go along. And another term we're going to use is prevalence. So it's like the prevalence of a disease or the prevalence of an infection. It just says how common it is in a population. So you can imagine this room of all of us being a population and like, what is the prevalence of the common cold right now? So if 5 people out of 50 have the common cold, then it's 10% prevalence, and that's just simply what the concept is. And then I want you to meet the bad guys of the story, although in their defense, they're just trying to survive too. But the major classes we're going to focus on today are bacteria, viruses, and parasites. And the Black Death is actually an example of bacteria. And it's the kind of thing that wouldn't actually be a big problem now, because what we have now that they didn't have in the 14th century was antibiotics. But that doesn't mean bacteria don't really still pose a serious threat, because there's a staph bacteria, and maybe some of you have heard of MRSA, which is like very resistant staph aureus that does not respond to a lot of the drugs we have. So that's something that, and when those bacteria made an adaptation to try to overcome what we're throwing at them as we're trying to stay alive, they're also trying to stay alive. And also another example is strep throat. Viruses are very common. There's HIV, which is going to feature a lot in this talk. There's also Ebola, which some of you might remember from the epidemic that occurred in West Africa a few years ago, and also something that's less epidemic, but all over the place, the flu and polio virus. And finally, parasites. Malaria is a parasite that Wes, Rebecca, and I study, and also going to feature heavily in this talk. There are also a lot of parasites that are actually in the shape of worms, and one example would be in worm disease, which is a place where we're actually really close to eradicating this parasite from the planet. There's like, last year I think there were 15 cases of a whole worm, which is amazing. And also toxoplasmosis, which is another parasite that a lot of people in the States might be familiar with, because it's kind of like transferred between human and cats. So if you love your cat and your cat loves you, you might have to. So those are the enemies. But going back to other examples of epidemics. So we had the SARS outbreak in the early 2000s where 774 people were killed. This is an example of a hospital in China where the outbreak was mostly localized too, but when you consider that at least 1/6 of the world's population lives in China, something being contained to China doesn't still mean it's not an epidemic. And then another example will be in 2009 when the swine flu started in North America. This is an example of how, these pathogens change our behavior because you have people trying to go about their daily life but trying to take steps to protect themselves. And if the pathogen wants to be more infectious, it has to find a way to overcome our defenses, and that's really part of the battle that we're really going to go into. And from there, it spreads like everywhere. A lot of deaths from this. Officially, 18,500. Well, this is suspected to be a low estimate, as many cases were suspected to be underreported. And most recently, some of you might have seen in the news in the past couple of years, the Ebola outbreak in West Africa that killed more than 11,000 people. This picture is an example of the burial protocol that they had to institute where, the Ebola is an interesting case in which once the person dies, they're still pretty infectious. Actually, they become like the most pretty infectious right after they're dead, and the virus can transmit from person to person that way. So thankfully, this outbreak is finally over. It actually ended right towards the end of 2016 officially. So that was wonderful to hear. But really the point of this is that, infectious diseases and pathogens are still a major influence on the way we shape our societies, the way we try to behave. And when they get to the point of rapid exposure, when they start to grow into epidemic proportions, they can really impact human life and behavior. But that's definitely not all in the past. So this graph is showing you global causes of death. This axis is of millions. And this is from 2000, at the beginning of the millennium. And you can see that tuberculosis, HIV/AIDS are pretty high up there. We have a lot of the chronic diseases, the killers of developed economies. But, and this is globally. So if you actually zoom in to low-income economies, and this is more recent data from 2015, you can see HIV, tuberculosis, malaria being really significant killers, and this axis is the same in 100,000 population. So these are not things that are just, we had epidemics in the past, and now we know how to take care of that, and we know how to take care of infectious diseases. We can do a lot more now, but again, going back to this concept of the arms race, they are trying to survive whatever we're throwing at them, and we're trying to survive their adaptations. So I will just give an example, dive a little deeper into two examples, starting with malaria. And malaria is what we call an ancient human disease. There's evidence of malaria infection from mummies, from in Egyptian mummies. So they've been around for a really long time. The most recent evidence we have suggests that the parasites passed from gorillas to humans about 10,000 years ago, which is a really long time ago. And pretty interestingly, I thought this was really cool when I discovered this. We found mosquito fossils carrying malaria parasites. Now, these are like not the human type malarias that would infect us. So you can think of malaria, but the parasites also infect a whole range of animals. So you can imagine that dinosaurs had malaria. prehistoric animal, so that's pretty cool. But it's not only ancient, it is still with us, definitely. So caused millions of deaths, hundreds of millions of deaths in the 20th century, and even last year caused more than 400,000 deaths every year. And the thing about malaria, which is what inspires me to study it, is that it is the biggest, the highest proportion of its victims are children under the age of 5. And I just think about all the things that those kids can contribute to the world that I never had an opportunity to do. And if we could find a way to decrease that proportion of deaths, then maybe we can solve climate change or all the terrible things happening in the world. So. And now switching to HIV, which is a more modern human disease. This sort of really blew up in the early 1980s, where it sort of started out in being noticed as a disease in the homosexual population, mostly in the United States and in Europe. And these are just a few headlines from then. And it's for a disease that is relatively recent, has infected more than 75 million people, and there were over 2 million new cases just 3 years ago. There was a lot of work being done for the control, but it's still a really big problem. And then I think one thing that I did want to say with relation to HIV is that even though it, I lost a slide. So it is a modern disease, not only because it really first broke out in the human population in the 80s, but actually doing a trace back of where the virus itself eventually came out of. It was in the 1920s in Central Africa. So HIV as a disease and the virus is pretty much like less than 100 years old, which is pretty crazy when you think about it. But it's something that is, so it's a virus, it's a different type of pathogen than the parasites of the bacteria that I was talking about. But really definitely still affecting a lot of lives. So what I hope I conveyed to you about this is that these diseases, these pathogens affect our population for sure, and in many ways they do affect our behavior. What we're going to get into now is how do they impact our biology. But before we do that, I will be happy to take any questions you have on this section. Oh, yes.
I have 2 questions. The first one is, the malaria is the transmission is through the mosquito, right?
Yes.
Oh, okay. And the second one is, when you show a map, you show like the countries with more deaths of malaria, and most of them was in the center of the globe. So I am wondering if it's because it's like a warmer, it's close to the Ecuador. You know, like as warmer, so the mosquito can grow better in that kind of environment.
Well, it does.
And the coldest, like here in Massachusetts, you know, it's so
yeah,
cold.
thank you for bringing that up, because you actually reminded me of a point I meant to say that.
Could you repeat the question?
Oh, I'm sorry. So the question is asking that when you look at this map of malaria risk, is localized to the middle of the globe, and it's wondering if it has anything to do with the location of the equator, which is actually right about here.
Because so many poor countries,
Yeah.
incidentally, in that.
So yes, thank you for asking that question, because one thing I meant to say on this slide is that while this is the current demonstration of malaria burden today, if you wind back this map to let's say 1900, all of the United States will pretty much be red, and parts of Canada up here, most of Europe will be red. So malaria was actually pretty endemic and all of South America will also be red. It was pretty endemic pretty much around the world, and actually since the 40s and 50s, there have been eradication campaigns in different countries to try to eliminate the disease with different different levels of success. One of the places where it was very successful was in the United States, to the point where even though you have mosquitoes in the United States, there is like really no malaria in the population, so they transmit it across. Whereas in a lot of many countries, you still have active transmission. Some of the countries that are red in Central America, I think El Salvador will be one of them, but there's not been any local transmission in El Salvador for the past 5 years. They have cases of malaria that show up there, but then usually trace it to somebody who came in from out of the country with malaria. So it's definitely, it's a very complex set of reasons, and part of it is because of the vector, the mosquito, as you mentioned. Part of it is something related to disease poverty. Issue of you don't have good health systems, good governance, and might exacerbate this. But yeah, thank you for another question.
Yeah, I'm curious about just definition of what constitutes an epidemic. Is it just incidence, or is it something like a rapid out of control growth of incidence? So I'm wondering, like, common cold, like everyone has it, everyone has had it, but it's not an epidemic.
Right.
Not worried about it. Like, why? Like, what is the line?
I think, I believe, and one of you want to join in with me on this. If you
repeat the question.
Sorry.
Yeah.
I'm so bad at that. The question is, like, where do you draw the line on an epidemic? Because the common cold, everyone has it. But what would actually describe the common cold as endemic? Because everyone has it. It's just in the population. It's sort of like a fact of life. An epidemic, sort of, I think the definition of like something that's like has a focal point. Like that's why they do the trace studies and then spreads out rapidly. I think one more question.
So you mentioned that there's this parasite, this guinea worm, that's almost completely eradicated from the world. I wonder if there are any approaches like that to malaria that just decide to eradicate mosquitoes entirely from the world. And is anybody pursuing that or talking about that?
The question is about the guinea worm example where it's almost eradicated. Is something like that happening with malaria? And is it possible to eradicate mosquitoes? Well, definitely we're talking very seriously about trying to eradicate malaria from the world. Actually, the team at the School of Public Health here as a defeated malaria initiative about 10 years ago. Bill and Melinda Gates got up and said they want to eradicate malaria from the world in the next 50 years. And they put a lot of money and resources and put like the weight of their celebrity and status to really go behind that. And it's really shifted the landscape. That said, I must say I'm optimistic in that I get up every day and I work on this and I try to do my part to make it a reality, but there's also the part where I don't think we can eradicate mosquitoes because they serve a pretty important point in the ecosystem. There are very cool studies, some of them going on right here at Harvard School of Public Health, where they're trying to modify the mosquitoes so that they're not able to transmit the parasites. That thing that's pretty cool. There are studies trying to infect the mosquitoes with a kind of bacteria that infects the mosquitoes, and once they have that bacteria, then the mosquitoes can get infected with the parasite. So there's definitely a lot of really cool and exciting work going on around that, and lots of people are thinking about it. But malaria has been with humans for pretty much as long as it's been like humans. It was with like our primary common ancestors. So there are times when I despair that we
I want to add really, really briefly to that because I think guinea worm is an example really worth discussing a little more. I think another really striking difference between guinea worm eradication and potential malaria eradication is that a lot of the work surrounding like getting rid of guinea worm in a lot of African countries has just been simply filtering the water. So the way that you get guinea worm is actually you swallow eggs that these little water fleas carry around, and if you don't filter your water, you ingest it into your system, and then you get that parasite once it happens. So a very simple solution is simply increasing sanitation. Because you can't really sanitize out mosquitoes from your environment,
yeah, and it's really impressive with guinea worm that they've done really amazing work because we actually have no medicine. against it, and yet it's almost eradicated, and it's all entirely behavioral changes. So another example of how these pathogens that really influence human behavior. And I have to wrap up to give my lab-mates a chance to tell you the really cool parts of this talk.
Well, while she's setting up the mic, having had them say all that, it is actually possible to try to eliminate malaria by targeting the mosquito, and that's exactly how it was done in the US.
Just kidding. Just kidding.
It's really hard to attach 3 mics.
Yeah, it's a lot.
I want to improve on that for the next section.
Yeah.
Alright, so I'm here to talk about general principles of evolution. And so the theory of evolution is basically the closest thing that in biology that we have to the unifying theory of everything. It is pretty much the best theory that we have for trying to understand why is it that we see so much diversity in life? Why is it that there are fish swimming in the oceans? Why is it that there are birds flying in the sky? It is also the best explanation that we have for trying to understand why certain organisms, in this case the finch, have different traits, in this case, beak size. But in this case, this is an example of one of Darwin's finches, and through a series of studies, he actually suggested that beak size in these finches correlated with their diet. Smaller beak sizes allow certain birds to be better at eating insects. Mid-sized beaks could allow birds to eat other types of bugs, and very, very large beaks to help with having the necessary skills to crush very, very hard foods, such as nuts and seeds. And it's also the reason why you are you. But how does evolution work? We have this idea that evolution is the cause of all the biodiversity that is observed in the world, but how does it work? How does it actually work? So in order to get there, we have to do a little bit of a genetics lesson. So please bear with me. At the heart of every single organism is a molecule of DNA. And this DNA basically tells the organism, this is the traits that you're going to have. And this, in this case, this DNA molecule tells this bacteria to be red and to be smooth. In order to reproduce, this bacteria must copy its DNA and send that DNA to its offspring. Sometimes the copying is perfect. When that copying is perfect, you end up with an organism that is identical to the parent. But sometimes it kind of screws up. When this happens, it's called a mutation. In this case, this blue guy inherited a mutation from the original red guy that now makes him blue. And this process can repeat over and over and over again, and resulting in greater and greater complexity. So from this kind of very simple example, you can start to see how you go from an organism with red cell wall and smooth body, down to an organism with a blue cell wall and lots of little hairy legs. But as soon as you start having differences in organisms, you open up room for competition. And the way they compete depends on a number of factors. It depends on the traits that they inherited or the traits that mutated somewhere along the line. These differences we call selective advantages if they're favorable, or selective disadvantages if they're disfavorable. Selective advantages can come in a wide variety of forms. Some of them can be as simple as, hey, I can find food better. And some of them can be a little bit more aggressive. In this case, the red guy beats up the blue guy. And does anybody know the ultimate goal of life, at least from an evolutionary perspective? I don't want to hear like, oh no, to drink Starbucks every day. That's kind of not what I'm looking for. Does anyone know what the goal of life is according to evolution? Procreation. Exactly. Leaving as many children creating as many children as you can. The reward of evolution is the ability to create children. And this is how, from a population level, let's consider this process on a population level. Let's pretend that in a population we have a single bacteria. In the first generation, it's going to leave some number of progeny behind, some number of children. Most of these children will be identical to it because DNA copying mechanism is actually pretty good. Sometimes we'll make a mistake, and therefore you have a mutant rise in the population. After another generation, you have more offspring being created. Now here's where selective advantages start to come into play. Because it is impossible for the population to keep growing infinitely. There will eventually be a limit. So if you have a pathogen in one organism that leaves behind more children, this population will eventually be overrun, such that we will all be red. But this is kind of a very simplistic approach. It's the selective advantages that allow for organisms to leave behind more or less children depends on the environment. And there are many, many ways an environment can alter these dynamics, such as in the case of drug pressure. In this case, let's say we are in a population that is pretty stable, but all of a sudden we introduce a drug in the population that kills all the red guys. Well, suddenly all these guys are going to die, leaving behind only the blue individuals to procreate. And now, after the drug pressure, we end up with a population of primarily blue individuals. And this is how evolution works at a very basic level. Does it work on humans? And how can infectious disease be a driver of human evolution? Well, let's take an example. In this case, we have this red pathogen here, and he really, really likes to infect and kill blue individuals. That's why they're all very scared. Because they can be infected, these individuals are considered susceptible. These green individuals are resistant to this disease. They will not be infected, and they will not die. Well, let this epidemic run its course. It wipes out all the susceptible individuals. But wait a minute. Wait, what happened? There's no one left to infect in the population. This pathogen basically killed everyone so quickly, there's no one left to infect, and it will eventually die off as well. After some time, these survivors will create children and repopulate the population, and maybe there's a mutation that happens somewhere along the line. This population will be completely immune to disease if, and only if, that red bacteria was the only guy out there. What if there was a blue guy out there? And this blue guy can infect these green guys. So you can start to see how we're in a tug-of-war situation between us and the pathogen. We are constantly trying to evolve in order to avoid being infected, and the pathogen is constantly trying to evolve to infect us. And this is where the concept of arms race is. What happens in the human population will cause changes in the pathogen population. And they're constantly trying to outcompete each other. So, I would like to shift gears a little bit, get away from this very basic genetics, and tell a little story. Does anyone like stories? How many of you know who Charles Darwin is? Raise your hand. So, how many of you are aware of his famous voyage on HMS Beagle to the Galapagos Islands? And how many of you are aware of how British society really liked his ideas? By the way, they really hated it. This is a caricature of one of the newspaper articles back then when he first published Origin of Species, and they could not believe it. How could an upstanding Victorian man be descended from the dignity? But alas, this is not the story I'm going to give you. This is a fascinating story. If you really want to listen to it, come to see me afterwards, and I could give you some great books that you can read about it and some more historical insights to this fantastic story. But our story doesn't happen in the 1800s. Our story starts in 1945 in Italy at a conference for population geneticists. Now, population geneticists are a type of evolutionary biologists. We, because I consider myself to be a population geneticist, are interested in studying evolution at the genetic level. So here in Italy, we got this conference full of very smart people, all of which are very passionate about evolution. One of them is named JBS Haldane. And he was one of the very first people to really start to put the link together between how infectious diseases can affect human evolution. He posited that for humans to survive, or for any organism to survive an infectious disease, there must be some level of genetic diversity already present in that population. Because when a highly specialized pathogen comes in, it will only be able to target certain individuals. Once the epidemic passes, well, once the epidemic passes, the population as a whole, because there was so much genetic diversity there, is relatively unharmed. Sure, it lost all the pink guys, but you still maintain like 90% of the people in the population. So he hypothesized that one way that humans have evolved in order to overcome infectious disease is to have lots of diversity, to generate traits that are very, very diverse. And for Haldane at the time, he hypothesized that one trait that was already known to be extremely diverse, that could fit his hypothesis, was blood. If you think about it, normal blood already has many different types. It is very diverse. There is A type, B type, AB, O, and you have RH plus and negative. Just for normal blood, as far as we know, these don't confer any selective advantage or disadvantage. They're also not associated with any disease. So when you start to consider the types of blood disease that are out there, though, you have many things with really cool names, like thalassemia, hemophilia, and like a ton of others. If you actually just like Google blood disorders, you end up with pages of like thousands of items. So for Haldane, blood could provide an answer for trying to understand human evolution. Simultaneously, and time history has lots of coincidences, simultaneously in Oxford, a young South African British doctor by the name of Anthony Clark Allison was also interested in understanding the role of genetic diversity in human populations. He was interested in generating a blood map of all the individuals in Africa and understanding what types of blood types are there out in the world. And he would eventually start to study one particular blood disease known as sickle-cell anemia. Yeah, anemia got chopped off. Sickle-cell anemia. So sickle-cell anemia, or sickle-cell disease, is a disease that primarily affects human red blood cells. And it's important to note that this is not an infectious disease. You cannot be infected with sickle-cell anemia. Sickle-cell anemia is a genetic disorder, which we'll get into later. The reason why this is bad is because normal red blood cells have to squeeze through these very, very tiny capillaries. But these sickle cells have a harder time squeezing through these very tiny capillaries and will get stuck. Once they get stuck, it causes a whole bunch of disorders within the human body, such that most individuals that have sickle cell disease eventually die. As I mentioned before, sickle cell disease is actually a genetic disorder. Unlike bacteria, humans actually inherit 2 copies of their DNA, one from mommy and one from daddy. For a normal person, you will inherit 2 strands of DNA that do not have a sickle cell mutation in it. You can also, because you have 2 strands of DNA, have one DNA that has the mutation in it and one strand of DNA that does not have the mutation in it. This condition is known as sickle cell, he's a sickle cell carrier. He carries the mutation for the disease, but he doesn't actually have it. It's not until you have 2 copies of it do you actually have the sickle cell disease phenotype and sickle cell disease. All right, so that was a big, long genetics lesson. Let's go back to history. At the time, something that was puzzling scientists was, how can sickle cell disease be so common? It's a disease that's killing people before they reach reproductive age, so technically speaking, they're not leaving behind children that have these mutations. Why is it so frequent? If you look at the distribution of sickle cell disease in the world, you notice that, there's a lot of it in Africa. And that was actually the prevailing idea at the time. They all believed that sickle cell disease was just an African thing. Maybe it's something that just African people just happen to have. Well, A.C. Allison didn't think so. If you look at Africa, you notice that there's a lot of heterogeneity in there. Some places have a lot of sickle cell disease, some places don't have very little of it, and some places have none of it at all. And this puzzled him. If this is an African thing, why is it not uniformly distributed all across Africa? And the more you interrogate this kind of hypothesis that is just an African thing, the more this hypothesis falls apart. Because You see sickle cell anemia in Greece and in India as well. And at the time, there were not that many Africans there, so let's just roll over that a little bit. By the time, it didn't make sense. How can you have a supposedly African thing in a place not in Africa? He had a couple of ideas. Well, maybe sickle cell disease is the result of rapid mutation. Maybe you have a normal individual and he just keeps accumulating mutations so quickly that he just leaves behind lots of progenies with double mutations. Well, after doing some back-of-the-envelope calculations, he decided no. The mutation rate will have to be 3,000 times faster than the no mutation rate of human beings. And to him, that seemed unreasonable. He took a little bit more look, and he started to realize sickle cell disease is highly associated with malaria. In areas where sickle cell disease is very prevalent, you also see lots of malaria. And he started to have this idea. Maybe sickle cell disease, or maybe sickle cell carriers actually, have a selective advantage. Those individuals that have the disease are dead. They're not going to reproduce. But maybe if you have only one mutation of it, it will be enough to provide you protection against malaria itself. With this hypothesis, he got to work. He scoured the African continent, looking at all the different blood types from many different African tribes, many of which were living in areas with malaria, and many of which were living in areas without malaria. And he also did something to me is incredibly cool, and also completely unethical. He somehow managed to find volunteers to be infected with malaria. It was a different time. We now know that today that his hypothesis is correct. Sickle cell trait, having only a single mutation of the sickle cell, having only a single sickle cell mutation is protective against malaria. As I mentioned before, people that have both mutations of it will typically die before reproductive age and leave behind no children. Individuals in an area with lots of malaria that are completely normal, have no sickle cell mutation, will have lots of parasites in their body and eventually succumb to the disease. Whereas those that have only a single mutation, sickle cell carriers, for some reason, we're not entirely sure why, but for some reason, they seem to harbor fewer parasites in their body, and they're able to recover from the disease much faster. Enough so that they will eventually leave behind children. All right, so did anyone else catch the little other niggling, something doesn't make sense here? Why is it then that sickle cell trait, sickle cell carrier condition, is not everywhere? Why is it that we still see normal individuals out in Africa, and why do you see individuals with 2 mutations in it? If natural selection were actually happening, wouldn't you expect everybody to be sickle cell trait? And here is again where biology is messy and genetics is complicated, and why am I studying this? It turns out the fact that we inherit one molecule of DNA from our mother and one molecule of DNA from our father is very, very important. Let's say you actually do have 2 sickle cell carriers that come together and mate to create offspring. They survive malaria, they're perfectly fine, and now they're wanting to make some babies. Well, the first child, one of the children they have, could inherit a normal copy of DNA from the mother and a normal copy of DNA from the father, thus generating a normal phenotype, a normal individual, a normal sickle cell, no sickle cell trait, no sickle cell disease individual. Alternatively, that child could be very unlucky and inherit two mutant copies, one from the mother, one from the father. And there are 2 ways of generating sickle cell carrier. In this case, this guy inherited the mother's unaffected copy, but the father's affected copy. In this case, this individual inherited the mother's affected copy and the father's unaffected copy. But you can kind of see from this very simple diagram how even though you have 2 individuals that are sickle cell carrier, they're going to always result in or have sickle cell disease. And that's why it's this is the reason for why in Africa you see all 4 of these, well, 3 types of this, 4 different genetic ways, 3 actual conditions. And that's why we have those 3 actual conditions floating around the population. This type of selection is known as, is a form of balancing selection or heterozygote advantage. Again, that's a very big word. Come talk to me afterwards, and I could give you many more cool examples of heterozygote advantage and some of the theories that arrive about from it. But Allison provided one of the first studies that provided evidence for Halding's hypothesis. And this is how science works. Some guy comes up with an idea. Normally it's the same guy, but it's consumed. Some guy comes up with an idea, and he or someone else will eventually go out and test that idea. If that idea turns out to be correct, then it's accepted by the scientific community. If it's not, then they're sent back to the drawing room and they have to come up with another idea. Normally it involves a lot of grant writing and money begging, but all right, so questions.
So that's what the sickle cell well, so if like one person has both sickle cell genetic they will die, but the person that has none will also die because of malaria. So basically there's a 50% chance of surviving.
So under this, yes, if you have those kind of probabilities where basically if you're normal, you're doomed. Yeah, sorry. He's specifically asking about, sorry. He's specifically asking about this, and he noticed that of the 4 children he positive had, 50% of them are expected to be sickle cell carrier, 25% of them are expected to be normal, and 25% of them will be sickle cell disease. So if you were to assume that malaria is really, really good at killing all the normal individuals, and that sickle cell disease is really, really good at killing, then yes, you would expect that 50% of the children that these individuals have will die before reproductive age. It's not quite that simple because even in malaria, even in areas with lots of malaria, some individuals with normal phenotype will still survive, and some sickle cell disease individuals will still survive long enough to produce children. So it's not quite as perfect as that, but it's very close. Well, it's a good thing that you caught it.
I think it's important to note too that although we used to consider sickle cell disease something that inevitably meant that you would die, like now there's really much better, more modern medicines where people with sickle cell disease can live for a much longer period.
I'll take the lady first.
You mentioned the different areas where the Africa, India, and Greece. Do you think that it's possible that this trait evolved separately, or do you see it as one something that evolved and spread out?
That's a great question. So she was asking, why is it that you see the mutation in Africa, you see the mutation in Greece, and you see the mutation in India? And there are 2 hypotheses for how this can occur. One is that some individual from Africa very long ago carried that mutation over there. It's just a migratory event. The other possibility is that it mutated spontaneously in Greece independently of Africa and independently in India. That type of evolution is known as convergent evolution, where you end up with the same trait, but the evolutionary path, the evolutionary history that led to that trait is different. And so that's a question that, as far as I'm concerned with sickle cell anemia, I believe it's independent instances of mutation. So I think it's an example of convergent evolution, but I'm not entirely sure. I might be wrong.
Yes, you stated that the prognosis for people with sickle cell is or the diverse approximately how individuals live with the disease, and on this map it's showing the eastern part of the world. What's sort of some of the rates that disease happens on the western part of the world?
Can you repeat the first half of the question?
Yeah, so approximately what's the prognosis on what age on the lives who have sickle cell anemia?
Does anyone know? I'm not a clinician, I'm an evolutionary biologist.
Could you repeat the question?
So he was asking, at what age does it become apparent that an individual with sickle cell disease actually has the disease? And I'm not entirely sure about that. I would have to ask the clinician for that particular piece of information. For the second half of it, what's going on in the rest of the world? What's going on in terms of sickle cell trait distribution in the rest of the world, such as maybe like North America, South America? It kind of depends on what time period you're looking at. Prior to the 1700s, you would expect to see there's pretty much no sickle cell anemia in that area. But after the 1700s, well, maybe 1600s. After the 1600s, so people started to see sickle cell anemia there as well. Can you hazard a guess as to why that happened?
Assuming people moving to
Yeah, exactly. It was kind of like forced immigration. So it's suspected that the reason why before the 1700s you did not see sickle cell anemia in the New World is because of the Atlantic slave trade, where Europeans were busing like boatloads of African people into the New World.
I have a question. I was watching a video on internet and YouTube.
YouTube is great.
And they were talking about some research they're doing with CRISPR-9 and like the DNA. So they were talking about I think they took the spermatozoid from men and they made some mutations.
Yeah.
And they took the egg from a woman, and when they put the sperm in the egg, and pretty much I think, I don't remember it was 100%, but pretty much the healthy egg from the woman fits on the DNA.
Yeah.
So that doesn't sound like a 50-50, right?
So his question has to do with gene editing. So recently in the news on YouTube apparently, by the way, what do you do for a living? Why do you have time to watch YouTube? Anyway, he's asking specifically about gene editing, particularly a technique called CRISPR-Cas9. And he actually knew a lot more about it than I was expecting. But it gets to this kind of inheritance. He said that in the video, they were able to take individuals like this. Maybe he has a mutation, maybe the mother has a mutation, the father doesn't have a mutation. But instead of a 50-50 ratio of people with, one copy of normal, one copy of mutant, they were able to generate progeny almost with 100% efficacy, progeny that contain either no mutation whatsoever or both mutations that they were trying to get. Gene editing is weird like that because it does violate this principle completely. That is why it's exciting. This problem of two DNA molecules has been dogging geneticists for a very long time because if you're trying to eliminate a mutation, you have to get rid of it from both DNA molecules at the same time. Before gene editing and before CRISPR-Cas9, it was very difficult to do that. You basically had to wait for 50, do probability 50% of the time you get it, 50% not. Gene editing allows you to get that mutation out of there or in there with 100% efficacy and violates this principle. The exact mechanism for how it works is a little bit too complicated for this talk, but feel free to come find me afterwards, and I will be happy to tell you.
I looked it up, and most infants that have sickle cell anemia are diagnosed right at birth, and even if they're not in a place that has very good diagnostic tools, and the diagnostic tools typically blood tests, as you can imagine, they'll start exhibiting symptoms by 5 or 6 months of age, and even very young children can experience really severe anemia and chronic pain, so it's really, really early.
Let's repeat that. All right, so is there any last minute question? Yes.
Since we saw that in the 14th century, humans died, and recently only 13,000 died.
So she's asked, are we winning this arms race? Are we actually beating infectious diseases? Will there one day be a society that contains no infectious disease whatsoever? And the question to that is yes. The answer to that is yes. The answer is also no. Because it kind of depends on what race you're referring to. If you're referring to the evolutionary race of humans generating mutations in order to overcome infectious disease, I think we're going to lose that. And that's because we as humans, on average, generate a single offspring once every 20 years. Whereas bacterium like E. coli produces an offspring once every 8 minutes, or 20 minutes. I don't work in the lab. Once every 20 minutes. So it's that difference in generation time that really causes bacteria and smaller pathogens like that to evolve much faster. They have more opportunities to mutate. But we humans are smart. We have drugs, and that is kind of like the one way that we're trying to overcome this disadvantage that we have. Unfortunately, for the same exact reason, because bacteria and other pathogens evolve so quickly, even these are starting to fail a little bit. So I am confident that one day we'll be able to eliminate all diseases. I just don't think it'll be very easy, and I don't think it'll be done in my lifetime. Right, and now I will hand it off to Rebecca, who will take it away from history and bring it.
So before we do that, let's actually take a 10-minute intermission. During this time, I want to encourage you to come talk to the speakers if you have further more detailed questions. Come talk to Dana if you want to ask us about things that we may or may not know the answer to. Grab more snacks and grab some flyers. I also do want to advertise the fact that this group is going to be leading a very cool lab tour through their malaria lab after this lecture. So if you would like to do that, and I promise you will not get malaria, there are only 10 spots, so we're going to put a sign-up sheet at the front, and please sign up.
Who are you looking for? It's like the first. Just like the first, like, the first time, right? Yeah, right. And we also like for tomorrow's event. Oh yeah, that is the third. Yeah, I can't come to that one. Oh my God, sorry. That's really fine. I'm really glad you're here tonight. I know it's not painless to hear, but that is okay. You just fell out of your bag. Yeah, so
Thanks for the photo, Nava.
Yeah, you saw the photo. Yeah, I did. Good job.
Thank you. I get so stressed out because it's like I just take a nap. Like I'm trying to talk, I take a nap.
When I posted the photo of you, I was thinking, oh my God, maybe I should have told every. I mean, if I knew like we were gonna do this talk, I should have like told everyone to come.
No. It's okay.
Because the talk is gonna be too like for like non-scientists too, so we didn't even say like everyone was saying that it's just a cheer for her, like the same thing.
Yeah,
Like if it's just like, as soon as we go back to is that what everything's good?
Good. No. I have 2 presentations tomorrow that I have to finish. Yeah, I'm finishing. One of them I have to email to my boss. Tonight, because they like to get the presentation. One of it, like actually the book, one of it's just an advisory meeting with my advisor. She's like finding out what I'm doing, what I'm planning on doing, what I want to do, advice. And the second one is I've been trying to do some coding that is not going to work.
Coding never works.
I hate coding. So basically we have this group meeting and basically as my boss was like, we just like use it as a workshop. You had this thing. I saw this on the bus. You know I made this logo.
Oh, you did?
Yeah, the fairy test. I did it. I didn't know they made a baby. I have to go get one now.
Because like we had like the leafness and like our boots like I'm in itchy ones too.
Oh, okay.
And our boots like right inside every single year like good, and I saw that and I was like so pretty. Can I click one? It was my first year.
Yeah. No, but I saw it because I saw it on the bus and then not on the pin, just the logo itself. I think they just put it on the pin. Yeah. I thought it was Tariana, but she had that something else. She had a bag that was kind of blue. And I was like, oh, is this your bag? Did you have the pin? And she's like, no, it's not me. And I was like,
I was if you have like a sticky like version of it, let me know.
Oh, I don't know. I just have the logo, and apparently you can just put it on anything.
I never like collect these things, but like after the Deadly Fest, I started collecting. I put like the GY on my computer. So
Oh,
I'm, like,
cool.
I'm gonna like hopefully like make these. Like,
I'll ask you about them if they want to do a sticky. That's a good thing to do.
Exactly.
Yeah.
So,
I should come. Like, I'm really terrible. So actually, like, I got a mentor through them. Like, the one I had last semester was amazing, and I'm gonna take with her this year. Like, it's really great. So I get all the emails, but like the events are just so like times that are just terrible for me. So like,
you should so you heard of the people so I was thinking
that might be good. She would love that. She loves interacting with students.
and then we can like plan and innovate and you can come too.
That's true. Yeah. No, actually, she's gonna be there. That's gonna like apply some right.
Okay, but it's like you can talk to her.
Yeah. That's a good idea. I will. She loves doing stuff like that.
Like you can like set it up for any situation you want. I know that you want.
Yeah. Probably here. Awesome. That's a good idea. I'll talk to you about it.
Yeah, I'm really curious. Like actually interested to see her.
Me too.
Oh really? You don't?
No, I did not see her. Just like
It's not very far along.
Okay, I'm gonna like email you probably like just for that because it's coming.
No, of course.
Sounds like really cool. Okay. Yeah, have you had like some?
I have some chance. Oh, yes, It's okay.
You sent me an email.
Oh my god, I love it.
If there were 100 people like this next to us,
That's why I sometimes been
Okay, if everyone is about ready, we'll get started with the final section of the talk. Okay. So we're going to finish off the last part of the talk, which is landmark battles. So we're going to dive a little bit more deeply into a couple of examples, learn a little bit more about two diseases we've already heard about, malaria and HIV. So here's a roadmap for the last part of this talk. We're going to start first off talking about a more molecular perspective of evolution. So Wes is a population geneticist. I'm a molecular biologist, so I think about things a little bit differently. We'll then go on, as I mentioned, just to focus on two examples. We'll look at malaria. We'll learn a little bit more about the biology of the malaria parasite. And then you already learned about sickle-cell disease, but we're going to get into the molecular basis of why sickle-cell disease happens and how, or what are some of the theories as to how this trait, or being a sickle cell carrier, protects against malaria. And we'll go into another example talking about how the parasite actually gets into our blood cells and how we could potentially make mutations to stop the parasite from getting in. And then in the last example, we'll talk about HIV. We'll discuss, so HIV is a virus, we'll discuss the life cycle of this virus in human cells, and then also how human cells can fight back and how viruses can respond. So again, let's spoke to you about evolution from a population level perspective. Now we're going to really zoom in, and so just to really start to zoom in, let's think about a cell. We already learned about red blood cells, that's one type of cell. But in general, cells are the building blocks of our body. Bacteria are a single cell. Our bodies are composed of millions and millions of cells. And cells, in my opinion, are beautiful. They have all of these different smaller components called molecules. So molecules make up our cells, and in particular, it's these molecules called proteins, which are mostly responsible for making up the various structures that you see in the cell and for the really important functions that are necessary for the cell to survive. So Wes told you that DNA makes up who we are as organisms, but more specifically, DNA provides instruction manuals for these molecules called proteins, which are found in the cell. And so proteins come in many different shapes. Oftentimes the shape of the protein reflects its function, reflects what it's doing in the cell. And so when we have mutations in DNA, these lead to subsequent changes in these different proteins, which are ultimately what cause changes in our cells and in the rest of our body that we can see. So those are the physical things, the traits that Wes talked about, like being a blue bacteria or being protected against a disease. So to understand how these changes in our cell biology can protect us against infections like malaria, we first need to know a little bit more about malaria itself. So you already learned that malaria is a parasite, and it came up, so that's a very good point in the question and answer session, that it's spread by mosquitoes. And as you may have guessed from Wes's lecture on sickle cell and blood cells, The malaria parasites spend most of the time when it's infecting us inside of our red blood cells. So a single parasite will invade our red blood cell and get in, establish itself, and then it goes about taking up nutrients, settling in, growing, and then after some time, about 48 hours, it divides into multiple new parasites. And these new parasites will then burst out of the cell and go on to infect new cells in the cell. This is a really cool video that actually came out of our department, just to show you what this process actually looks like under the microscope. So right here, this is a red blood cell, and this cell has been infected by a malaria parasite. You kind of see these little segments, like big segments. Each of those is a little malaria parasite that's divided. The dark kind of dot in the center is actually waste products that have built up as the parasites and taking these nutrients from the cell. So keep an eye on this cell here, and you'll see the parasites burst out, and now they're going on to search for new cells. Get them over time because it happens really fast. So you can imagine that having your blood cells being like burst open and then more cells getting reinfected and bursting again is not particularly good for you. And it's this invasion and bursting of red blood cells that leads to the symptoms of malaria. So you can have problems like anemia or low blood pressure, which you might expect. Another problem with malaria is that the parasites often cluster in the blood vessels around various organs, such as the brain, the liver, or the muscle. And this is often the cause of the severe disease because it leads to organ failure. So this is actually an image of a mouse that has been infected with malaria. And this is a really cool image because you can actually visualize where the parasites are. So the red and yellow colors indicate a bigger number of parasites in those areas. And what you can see from this image is that rather than the parasites being spread throughout the body of the mouse, they are really clustered or localized to particular organs, like the lungs, or the spleen, or the fat. And this phenomenon that the parasites cluster around particular organs is known as sequestration. As I mentioned, that's really important for the disease, especially the severe disease that we see. So now that we understand a little bit more about the molecular biology of the parasite, let's think about how being a sickle cell carrier could protect us against this parasite. So you already learned from Wes that with sickle cell disease, it causes your cells to form that crescent or sickle shape. But what's actually happening here on a molecular level? Well, we talked about how our cells have proteins. And one protein that's especially common in red blood cells is a protein known as hemoglobin. Hemoglobin is really important because it actually binds and carries oxygen. So it allows our red blood cells to carry oxygen through our bloodstream and around our entire body. So when we have a mutation, as Wes mentioned, it's a mutation in our DNA that causes sickle cell disease. And what this mutation does at this protein level is it actually changes hemoglobin so that it becomes kind of sticky. It causes these proteins to stick together in these long strands. And it's these long strands of the hemoglobin that actually cause the cell to become rigid, kind of collapse into that crescent shape. Now, but Wes told you that people with one copy with the sickle cell mutation and one copy with the normal, not mutated DNA, don't have the disease. And this is because people who have one mutation and one lack of mutation, what their cells look like is something like this. So they have a mixture of normal and abnormal. Cells that have a mixture of proteins generally are normal, except under conditions of certain stress. And so that's why people who have the two copies with the two types of proteins are really healthy. So how does sickle cell disease and what we know now, how does the presence of this abnormal hemoglobin protein help protect our cells against malaria? Well, unfortunately, I'm going to have to tell you that we don't actually know. This is something that scientists have been interested in for a really long time, and there's multiple theories that have attempted to explain it. But overall, this is an area of research that's really still ongoing. So at first it was thought that, and perhaps the most simple explanation is maybe just having a more rigid cell makes it more difficult for the parasites to invade within for that first step of the life cycle. But as it turns out, and as you might remember from Wes's talk, people with the sickle cell mutation, people who are sickle cell carriers, still can get infected. They are just more protected from the severe form of malaria. And it seems like when we look at people who are sickle cell carriers, the parasites are still getting inside the cells. Another possibility is that this mutated hemoglobin reduces the replication of the parasites, the ability of the parasite to grow and divide within the cell. So it's thought that maybe that it makes it more difficult for the parasite to access the nutrients inside the cell that it needs. Another theory which has some support from biology is that although the cells with both types of protein look normal under just normal healthy conditions, when the malaria parasite invades, it puts some stress on the cell. Sometimes that stress actually causes the cell to sickle. And then these collapsed cells might potentially get cleared away from the body, and along with it, clearing away the parasites. And a third theory is that the sickle cell mutation might reduce that sequestration that we talked about, so the ability of the parasite to cluster near different organs. And that would definitely be one explanation as to why patients who are sickle cell carriers are less likely to get those more severe symptoms. So these are some of the theories that have been tossed around, but the fact is that there are other theories, other possibilities that scientists have suggested, and it might end up being a combination of different things, as well as other possibilities that we didn't know about yet. So now that we've talked a little bit about the molecular basis of sickle cell and how it provides us protection, let's consider what other red blood cell traits or characteristics might also be able to protect us against malaria. So if we go back to the parasite life cycle, you can imagine that one really effective way that we could just stop this cycle is by preventing parasites from getting in the first place. That would be great. So let's look a little bit more closely at what's actually happening when the parasite invades our cells. So if you zoom in onto the edge of a red blood cell, you'll find that there are many different proteins which are known as receptors. And you can think of this, receptors are found on all of our cells, and they're really important because they're like our cells' communication lines. They allow our cells to receive messages from the outside environment, and these messages actually usually come from the other proteins, which come in and fit really specifically within the receptor, kind of like a key into a lock. Now, the parasite is really tricky. It takes advantages of the cell's receptors by making its own proteins that look really similar to the key that is supposed to fit into the receptor. This allows the parasite to come in and attach to the receptor within the blood cell. You can think of the parasite like a very clever bandit. It's taking the lock, but it's managed to somehow make almost an exact copy of the key. Just like a lock and key, this interaction between the parasite and the red blood cell receptor allows the parasite to break and enter into the cell, where it begins stealing very precious treasures, namely all the nutrients that it needs to grow, replicate, and make more parasites. Now, one way that the cell can avoid this is if there's a mutation, so change in the DNA, that causes a change in those red blood cell receptors, so change in DNA, change in protein. And this effectively changes the lock, so the parasite can no longer get in. However, as you know from Wes's section, we're not the only one in this evolutionary battle. The parasite can also make its own mutations. And so you might imagine that it's in this scenario, the parasite could get a mutation, And this mutation would change the parasite protein so that they can now fit inside the receptor again, once again copying a key that can unlock the cell. So in addition to changing its receptors, there are some mutations that can happen that actually get rid of this particular receptor on a red blood cell altogether. Essentially, and this preventing the parasite from getting in. Essentially, the cell goes into total lockdown. And there's actually one really cool example of this in the area. To understand that, I have to mention that one thing we haven't talked about so far, there's actually two main types of species of malaria parasites that mainly infect humans. And if you want to know more about them, definitely come talk to me afterwards. But for now, we're going to call them species A and species B, just to make things simple. So one difference between species A and species B is that they have different distributions. So these maps, the warmer colors indicate higher prevalence in those areas. And you can see that species B is pretty widespread across the African continent, South Asia, and some of South America, whereas species A seems to be restricted to certain areas of the map. It's interesting. Now, another difference between species A and species B is that they each use different receptors on the blood cell. So, two different species, two different receptors, and then two different sort of proteins the parasite uses to fit into those receptors. And there's actually a mutation that people can get that gets rid of, it doesn't get rid of these receptors, but it gets rid of these receptors that species A needs. And as it turns out, there's actually a really high percentage of people worldwide that have this mutation where they lack this receptor. So this is a map showing the percentage of people lacking the receptor for species A. And you can see it's largely within the African continent. In fact, in some areas of Africa, 99% of people have this mutation, but they don't have this receptor. And you might notice that the areas of the world where this mutation is really common correspond really well with the areas of the world where species A is not present. And this makes sense because it means that species A is restricted from these areas, and this is exactly because most people in these areas don't have a receptor that species A needs to get inside their blood cells. So I think that these examples, both sickle cell and the receptor example, really highlight how malaria has altered the evolutionary history. And it speaks back to Lola's section where she told you about the tremendous impact that malaria has had on the population over time. These parasites have been with us since almost as long as we ourselves have been in existence. They kill, the disease causes a lot of deaths, particularly in young children. So so people who have these different characteristics that make them resistant, that protect them against this disease, are at a huge survival and reproductive advantage. And this is really reflected in the high prevalence of these different mutations that protect us. It really speaks to the impact this disease has had. So move on to our last example, looking at HIV in evolution. So this was touched on in Lola's section again, but HIV is a virus. It's a virus that causes AIDS. And just a little bit of biology of the virus in the AIDS cells, whose normal job in the body is to fight infection. And this is the reason that AIDS patients have a weakened immune system and become unable to then fend off other infections. This is a typical life cycle for many viruses, including HIV. So first what happens is the virus has to enter the cell. What I'm not showing you here is that this process actually requires a really similar interaction between the virus and receptors on the cell surface, very similar to the parasite. And the other thing about viruses compared to other pathogens like parasites and bacteria is that they're really small. So this is not the scale. In reality, virus is about, the HIV virus is about 1/60th the size of a human cell. So the virus is carrying a light load. It doesn't have a lot of proteins. very simple. But viruses are incredibly tricky because what they do is they actually take over the cell's own machinery and they force the cell to turn into this virus-producing factory. So the cell will be making all of these virus proteins, the new parts for the virus. And then in the case of many viruses, including HIV, these virus proteins assemble at the edge of the cell membrane, and then of the edge of the cell, which is known as the membrane. And from there, they'll bud off and go on to infect other cells. So this is sort of basic virus life cycle that applies to a lot of different viruses, not just HIV. Now the viruses may be pretty cunning, but our cells have some pretty cool tricks in their own. One really cool example of this is a protein called tethering. Tethering binds to viruses as they are ascending at the surface of the cell, and then, as you might be able to guess from their name, when the virus tries to bud off, the tethering protein literally tethers the virus to the edge of the cell, which prevents it from going on to infect another cell. And then the protein even forms these long chains of viruses where it links multiple viruses together. This is known as what we think of as an antiviral factor. So it's a protein that's present in our cells that interferes with the virus. It's one of many examples, but I think it's a really cool one. Here's a really striking picture of what this actually looks like with a high-powered microscope. So at the bottom of this image, this is actually the edge of the membrane of the cell, with the sort of light gray. And each of these tiny dark circles is a virus. And you can see that they're all really attached to the edge of the cell. And then here you can see a close-up of some of these long chains that are forming. Now, as it turns out, this really cool tethering protein was first discovered in HIV research, but it actually inhibits a number of other proteins like Ebola and dengue. But something that you might be asking is, if we have this really cool defense against viruses like HIV, why aren't we resistant? Why do we still get infected and have disease with these viruses? Well, unfortunately, this is where we get back to that concept of the molecular arms race. It's a back and forth. And viruses, in this case, have their own counterattack. Now, different viruses have different ways of doing it. But in the case of HIV, which we're focusing on, it makes this so again, viruses still make a lot of proteins. Everything it makes has to be really important. And one of the proteins that the virus makes is this protein called VPU. It stands very originally for virus protection unit. And the main purpose of this protein is to bind up tethering and prevent it from attaching to the body of viruses, allowing the virus to go off. So here again is that photo that I showed you, but what I didn't tell you is that the viruses in this experiment were actually genetically engineered so that they don't have that VPU protein. And the same experiment, but where the virus does have the VPU protein, the virus is able to freely float away from the cell. So I mentioned tethering is what we call an antiviral factor, and there are other examples of antiviral factors. A lot of them actually work here to prevent this production of virus proteins. But just as in the case with tethering, for every antiviral factor that we discovered, as it turns out, HIV has come up with a way to fight against it. Now, you might remember from Lola's talk that HIV is actually relatively a relative newcomer to humans. HIV spread to humans about 100 years ago. So what I want to point out is that this molecular arms race is actually an ancient battle that's been going on throughout our evolutionary history. In fact, it's believed that tethering actually evolved in early vertebrate species over 30 million years ago. And it's thought that antiviral proteins like tethering protected our non-primate ancestors, like these guys, from more ancient viruses that are no longer around today, but which have made an impact on our DNA. So, Scientists have found that there's been, or they kind of suspect, based on genomic studies, that this virus has undergone a lot of changes over time. These proteins have undergone a lot of changes over time during our evolutionary history that really reflects this battle. So, we develop, or we get tethered in order to, or our ancestors get tethered to protect us from this virus. The virus has a response. It creates this UV protein, UV protein, and then this gains a mutation that allows it, that protects it from tethering. Tethering might be another mutation where it can now no longer run this and can still restrict viruses. So the virus has to make another mutation. So this can sort of attack this tethering, and so on and so forth. These two proteins are constantly changing, sort of upgrading over time, so that they continue to stay one step ahead. It's again the idea that we want to protect ourselves, and the virus in turn must change so it can infect us. So this is a perfect example of what we sort of introduced at the beginning of this molecular arms race. We gain a defense against the virus responds by countering it. We then change our defense, and the virus changes its counterattack. In order to strive, we have to keep constantly upgrading our defenses. But again, this is something that happens not only over the course of human history, but even as far back as our non-primate ancestors, and even before. So as an overall summary of our talk, so we heard from Lola that infectious diseases have had this huge impact throughout human history. Then from West, we really got into the nitty-gritties of evolution and natural selection, and how during evolution these advantageous traits are passed down from generation to generation. It's sort of what determines who makes more children, determines what our population looks like over time. Wes also discussed with you how pathogens can be a selective pressure, favoring traits or characteristics in the human population that make us resistant or protect us against disease. We got into the really cool example of the sickle-cell trait, which provides us with protection against malaria. We talk about how this really explains the prevalence of sickle-cell carriers and sickle-cell disease in places where malaria is common. There we started to get more molecular understanding of evolution and try to really an understanding of how changes at the protein level can potentially be protecting us from these diseases. As another example of this malaria, we talked about sort of the lock and key model, and use that as an example of how we can make a change and the parasite could potentially change in turn. And then finally, we discussed HIV and tethering as an example of this ancient molecular arms race. Now, before I take questions, I want to conclude with a little bit of a look forward at where we're going from here. So at the very beginning, Lola brought up epidemics like Ebola, and I think that kind of points out that we're still dealing with these old enemies. So things like malaria are still really common, and we still have a lot of work to do. But also there are these new challenges ahead. In the news, you might have heard of like Zika virus and Ebola, and many of these diseases are emerging diseases that we think spread from animals to humans. For example, the most recent Ebola outbreak was believed to originate from an infected bat. So one concern from these emerging diseases that are coming really out of the wildlife population is that we are evolutionarily unprepared to defend ourselves. Because diseases like these have only been in the human population for a short period of time, we haven't had any time to really evolve protections against them. And it's thought that this might be actually one of the reasons why diseases like Ebola are so deadly within humans. But I do want to end, of course, on a more optimistic note. We talked about how the question was brought up, are we winning or losing this battle? And as I said, it kind of depends. Evolutionarily, we're at a huge disadvantage, but on a more optimistic note, the deaths due to infectious diseases have been declining dramatically over time. And this, as we talked a bit about during the discussion, is really largely due to public health efforts, things like drugs, vaccines, and public health efforts like water sanitation or water filtering, as in the case of leukemia. So that was brought up during the question and answer session. But I also want to pitch that perhaps if we can understand the molecular basis, so understand what's going on with these natural resistances we have, understand what's going on with sickle cell, what's going on with tether and BPU, getting this type of research and getting these insights into our molecular biology could potentially help us come up with new treatments or new therapies in the future. This concludes our talk, and I'll take any final questions.
I was wondering, in the case of
yeah. Yeah, that's a great question. So one thing I'll say is that, you know, the pictures that I'm showing are pretty simplistic because, in fact, our cells have many different types of receptors. So in this case, my understanding is people with these mutations are relatively healthy, and that's partly because, you know, within our biology, maybe we have receptors that can fulfill multiple receptors that can help fulfill the same functions. But it is a great point. It's a reason why maybe we can't lose the receptors for the other species because it's too important. We have some limitations to the changes that we can make because, exactly right, these proteins are in our cells for a reason.
I'll venture one. Is there possibly a kind of tragic irony at work here in that the more our hardiness as a species depends upon the vaccines we can create, drugs we can develop to fight diseases, in fact, the less hardy we are as a species because we need these drugs and vaccines to stay alive, and therefore we are more susceptible to disease if suddenly we don't have these drugs. And the pathogens are getting cleverer and cleverer at finding their way through it.
I think that's an awesome question, which probably requires like a more complex answer than I even fully know about or that we have time for. But some things that come to mind, you know, I think it might depend also particularly on the particular pathogen. So with some of our public health efforts, we've been so successful that we can really start to, like in the case of guinea worm or smallpox, we can really start to completely get rid of these pathogens with our vaccines or drugs or other methods. And so I think in that case, you know, we can kind of get rid of the bacteria or the virus altogether from the human population. Then there's not as much risk of them coming back. There are some really interesting thoughts that, like, as infectious diseases, and this is actually something that you may know a little bit more about, but like, as infectious diseases have gone away from our population, some of these adaptations that we originally made in our early history are no longer advantageous and might actually put us at a disadvantage. So there's some really interesting literature on that, like the high hygiene hypothesis.
On the flip side, there's another perspective you could put at it. So without infectious diseases, you might expect the human population to explode. You're suddenly creating lots of individuals that can survive long enough to reproductive age. With every reproductive event, you have a mutation opportunity. So as the population is expanding, you're generating more mutations than it was before. And since these are new mutations rising in the population that have never truly been tested, some of those mutations could actually come potentially against disease. This is just a hypothesis, and it's never been proven before, but it's kind of a neat thought experiment just to see what is one possible direction that removing the pressure of infectious disease could either help us or harm us later on if another infectious disease would come back into the population.
Also add that pathogens are evolving way faster than we ever could. So even if we didn't have, you know, vaccines or drugs to kind of help us, our innate ability to resist them, I think, is very limited. So, well, you're right that perhaps like using vaccines and drugs is limiting our ability to evolve these protective capabilities, I don't think we can evolve fast enough to ultimately protect ourselves at all. So I think our advantage as humans is being, smart enough to kind of figure them out and try to get one step ahead, which, as we've seen with like Ebola and new outbreaks happening, is almost impossible. But that's why we need great science to keep trying to get one step ahead.
Right.
I'm wondering, what's the difference between viral infections and bacterial infections? Like, we're getting good with antibiotics, but not so good at fighting viral infection. What's the difference?
Sorry, I picked that up. So the question was thinking about viral infections versus bacterial infections and why it seems that we have maybe more drugs against bacteria. And what are the differences? So that's an awesome point. I'm so glad you brought that up because it brings to the fact that we have this big kind of class of drugs that we call antibiotics, and those are particularly drugs that act against bacteria, so they're not effective against viruses. And as to why we have so many more drugs against bacteria, I'm not sure if there's a great answer to that.
I maybe have a partial answer. So, it is true that we do have a lot of antibiotics, with the caveat that there are a lot of bacteria out there with resistance against those antibiotics, so we're not really necessarily winning that war either. But a lot of the antibiotics we have today were actually isolated from the environment, ironically from other bacteria that make the antibiotics to fight competitors for nutrients, for space, and basically duking it out between different forms of bacteria. And so we've been lucky in that we've discovered a lot of these things without actually have to make them ourselves. The case is not quite the same for viruses. So that's one big reason. It's not the only reason, but that's a big contributor.
I don't know if this is an answer for sure, but if you think about the idea that bacteria are much more complex, they have sort of a lot more going on, you might think that there's also like more targets to hit within a bacteria than within a protein, or within a virus that has so fewer proteins. But again, I'm not sure if that's an explanation, but it's an interesting thing to think about how the biology might really relate back.
Right.
Sorry, I just wanted to ask, how good are we being able to predict the next
That's also a great question. So the question was, how good are we at predicting the next outbreak? I think there's definitely a lot of efforts around that. Like there are efforts to actually go out and survey the wildlife populations. So like go into like monkey populations and look at patterns also where there's places where there's humans and animals, wildlife that are starting to live in closer and closer proximity. And so I think efforts like that to try to predict where diseases might arise out of animals. And there's like, I know there's different sort of mathematical models and to look at where there might be potential hotspots within the world. But I would say, like, there's still a lot of uncertainty for sure. No one predicted like the Ebola outbreak, and no one predicted that when it happened it would spread so much. So I would say we're still often being caught off guard, and I think it sort of brings up the point that we need to make sure that we're continuing to put research into viruses and bacteria, even if we don't think they're a big problem now, just in case they could be in the future. When Ebola hit, we didn't have a lot of good, vaccines or medicines or ways to fight it, and the scientific community really had to sort of struggle to put together everything they had to try to combat it. And so, using scientific research and serving the wildlife population to kind of prepare for whatever might come, I think is super.
Yeah, just to add into that, so what we are able to do is like with global health, like reporting systems are actually pretty good now, especially compared to in the past. And for example, the identification of Zika as epidemic and infectious one was something that was sort of like, as it happened in real time, was because of all the networks of research centers that are really all over the world, like, talking and discussing and bringing up their cases and really being able to get to what the root of the problem is, and we don't really have a solution yet. So, but again, just that it's all about keeping to do the research. Call your congressperson and tell them to give us more money. But it's really because these pathogens, the ones we've talked about and the ones that we've nobody has discovered yet, they have a lot of advantages on us, and we have to keep doing our best to make sure we get past them. At least until we can make children, and then keep them home.
All right. I know there might have been one or two more questions, but in the spirit of keeping on time and making sure there's time for the super exciting lab tour that's coming, I think we'll close here. Obviously, if you have any questions, come up to us after the lecture, but I do want to make sure we all give them a huge round of applause before we break.
Okay, so okay, unless you like to. Well, I would like to, but yeah, no, live your life. Cool, so we just send them off. I'll get them all this.
Thank you, Doctor.
You guys got any questions? Yeah. You guys got extra medicines? Cool. That's awesome. All right. Should I stop the recording? This one? All right, stopping.